Hi, and welcome to this next tutorial on the use of the Pecan plugin. In this tutorial, we're going to consider how exactly we can analyze images or two separate regions of two separate regions of interest within an image. So for this experiment, we're going to look at sample image 4, and what you can see here is we have another wing disk, and this time it's carrying a autophagy reporter called reflux. Make it a bit clearer. And although it's not explicitly marked as such, what we have is two separate genotypes within the same tissue. As you can tell, this half over here has lots of green speckles, indicative of a decrease in autophagy flux, whereas this side over here has much fewer green speckles. And that's because this half over here is expressing a RNAi, and this half over here is the wild type. So what we can do is actually analyze these two separate compartments within the same image. If these compartments were marked really clearly, like if there was a separate channel that was red versus green, and you could really easily mark it out, well, you could just analyze it as normal and just have some sort of way of segmenting out whatever the color is. But because we don't have a marker, we just have to draw it ourselves. But doing that's pretty easy, and I'll show you how that's done. So launch Pecan. And we still have our settings like up and running from the last example image. And let's say that let's imagine a scenario where you've been, optimizing some conditions for a analysis that you've done, and you found conditions that you really like. Well, you can just save them for later so that you don't waste your time or you don't have to write them down. So go down here and click Save Current Settings. Change that to example 3. That's okay. And now you'll see those settings are saved down here. So if we go back to the default settings, we're now back to analyzing, say, clones in a wing disk. We can go back to example 3 and everything is restored for us. So let's go back to the default settings. And we don't have any cell death in this experiment, so let's turn this off. And we're going to do manual selections again, because it's a wing disk and it's a bit tricky to automate that. But we don't have any clones, so we have to change this. So what we're instead going to use is use ROIs as clones. So whatever ROI we draw is going to be automatically converted to being the clone. And if we want to do two separate regions, so you can use this approach to do, if you have one sample that's all the same genotype or condition, and you want to analyze just all of a given region of interest within a particular sample, you can just do it this way, and you'll have one region of interest, and you'll just measure it that way. Or if we want to do two ROIs within a single tissue, click on Advanced ROI Segmentation Parameters, and we're going to do Compare two regions of interest in an image, and press OK. And also, the analysis we're going to do is fluorescence intensity and speckle analysis. So there's a couple. Normally it's disabled. We can count the number of speckles, or we can also get the count, the area, the size of individual speckles, and the fluorescence intensity of the speckles themselves. Let's do the full speckle analysis. And that was channel 2. That all looks right to me. Another thing that you can do is when you're sort of troubleshooting your conditions, because usually when you do the speckle analysis, you have to play with this number down here. So we're going to have to troubleshoot it a little bit. So to make that a bit easier, go on Save and Display Options, and tick this box, Display Output Panel. That just means that this the output image that we generate and normally take a look at is going to be automatically displayed when the analysis is complete, just to save us a step. Press OK. And go to example image 4. And so this is going to be the same process as normal. We're just selecting the first region of interest. It doesn't matter which you choose. It just matters that you're consistent about it for your analysis. That, for whichever, if you have two separate genotypes in an image, just be consistent about it. Otherwise, your measurements will be kind of backwards. So let's start with the wild type, which tends to be based on anatomical landmarks in the signal. It kind of looks like it's coming from over here. And that looks good from, let's say, 25 to 12. And of course, you can add more. You can add more z-planes if you want. But I think this is good enough for now. So just click Finished. Now it's going to ask you to select your second region for analysis. That's going to be this one over here. And let's just make sure that it's good over the range that we looked at. So 12 to 25. Okay, I'm happy with that. And just press OK. Okay, so the analysis is done. Let's take a look and see how it did. So as you can see, we have the two compartments that we've drawn. This will be compartment 1 and this will be compartment 2, just based on the order in which you drew them, and that's how it's going to be output in the data sheet. So let's take a look, and you can see that the ROIs we drew were just used as the clones. The border and center region still applies, but you can just kind of ignore that fact if it's not really relevant anymore. For the speckle analysis down here, we have on the left-hand side just the raw speckle channel that we're working with. And this middle one is an overlay where everything that is interpreted to be speckles. This is kind of showing you the area that is occupied by the speckles. So you'll notice that some of these are a bit bigger, and that's because if you choose to do area instead of just doing the count, there's going to be a minor seeded region growing step where it's just going to expand to fill up the entire speckle. And let's take a look over here where we have just an overlay where we've kind of drawn circles around everything we're counting as a speckle. So that's a speckle, that's a speckle, that's a speckle, that's a speckle, that's a speckle. And so whenever you do the speckle analysis is the most particular one. It's sort of the one that needs usually the most troubleshooting. But right now it's done pretty well, I would say. There's obviously a cutoff when you're doing speckle analysis. For instance, it's not counting that as a speckle, presumably because it's a bit too dim. But on the whole, I'd say it's doing a pretty good job. If you want it to be more sensitive or less sensitive, you can adjust that. It's one number. Right here where it says speckle noise detection tolerance, you can increase or decrease this to change the sensitivity of how many things it's going to count as speckles until you get something that you like. The speckle analysis is going to work on pretty much any sort of speckle staining you have that's good quality. So it's not going to really work very well if you have relatively high amounts of background or if it's not a particularly good staining. The reflex tool is really pretty good. It gives very nice sort of signal to noise, so you can obviously do the speckle staining on that. That's what it was designed to do. But I've also applied it to different things and different sorts of stainings. And that's how you can run the speckle analysis in Pecan.